Hi, everyone. As John mentioned, my name is Ronald. I am a postdoc in the class group here at JCVI. And today I'm going to be telling you about some of the work that I've done since I've started as a postdoc. So a lot of the work that I do as a postdoc involves collaborations with computational modelers and mathematical modelers of our minimal cell. So we have some collaborators that have established a model for how the cell may replicate its DNA. And what we want to do is design experiments that can validate a lot of these models. So in regards to DNA replication specifically, I'm going to tell you about a little bit of work that I've done trying to add a fluorescent label onto DNA, which is the major replication initiation protein across all domains of life. I'm going to talk to you a little bit about HUP-A, which is a protein that encodes for a histone-like protein in bacteria. This is a protein that condenses DNA to form chromatin in a manner that is similar to what we see in more complicated eukaryotic organisms. And then John alluded to this a little bit earlier today. We have a cell, JCVI SYN3A, that doubles every 2 hours, but we have a metabolic model that states that in order for the cell to double every 2 hours, it must have this enzyme called the transaldolase. Without this enzyme, the model predicts that the cell should double every 8 hours. There's nothing in the minimal cell that looks like a transaldolase. So I'm going to talk a little bit about some work that's been done to try and identify such an enzyme and how we can potentially characterize it. So I'm going to start by talking about DNA-A. What I want to show you first is some micrographs that were generated by Ji Wang, who is a graduate student at TJ Haas lab at Harvard University. These are our microscopy collaborators. What you see here is DNA in blue, the cell membrane in green, and FTSZ in red. FTSZ is a protein that forms a ring in most, but not all bacteria when they are ready to divide their DNA. In natural bacteria, what you typically see is a single layer of FTSZ fluorescence in the middle of the cell, which signals the plane of cell division. What we see in the minimal cell is multiple rings of FTSZ within the cell, suggesting that there might not be a defined plane of cell division in the minimal cell, which is in contrast to what we see in natural bacteria. Before cells fully divide into two daughter cells, the cell must replicate its DNA. That DNA is usually in line, is always in line with the plane of cell division, because this data suggests that there might be multiple planes of cell division in the minimal cell. We don't know where the cell is initiating DNA replication. So what we want to do, what I've been working on, is trying to design an experiment that allows us to simultaneously visualize cell division and DNA replication in the same cell. What I've done is add a green-red photoconvertible fluorescent protein known as M-MAPLE3 and fused to the C-terminal end of FTSZ. To look at DNA replication, we put M-cherry in the middle of DNA-A based on what's been published in the literature on DNA-A labeling and using AlphaFold prediction tool to determine to predict structures for DNA-A labeled to M-cherry. We specifically put it between residues 105 and 106, which is a rather unusual spot. The idea is to install these fluorescent fusions into the landing pad of the minimal cell, and then using super high resolution fluorescence microscopy with our collaborators, we hope to visualize cell division and DNA replication in the same cell. Unfortunately, labeling of DNA has not been very successful. So what I've tried so far is expressing a labeled DNA molecule under promoters of different strengths. When we do this, we are not able to recover viable bacteria, suggesting that overexpressing DNA with the label is potentially toxic to the cell and poisonous to DNA replication. The literature suggests that co-expression of DNA N, which encodes for the sliding clamp of DNA polymerase, may compensate, may provide viable cells. If you overexpress DNA, that did not work in our system. P Michael 1 is a replicative plasmid that can be used to that can be used in mycoides, and it also has DNA. If we propagate P Michael 1 into the minimal cell, we are able to get colonies. But if we stick a label in the same spot onto DNA on P Michael 1, we get no colonies. And if we swap out M cherry for GFP, a smaller version in a split GFP experiment, that also doesn't work. We consider the possibility that M cherry may be too big, but if we swap it with the fragment of GFP, we get nothing. If we substitute the promoter for a much weaker one, we're able to recover colonies, but it's not enough to give us fluorescence if there is any fluorescence. So we've thought of a couple of different alternative ways to look at DNA replication in a minimal cell. Nika is a postdoc in our lab who came up with this really brilliant idea for looking at DNA replication in a way that does not directly involve looking at the origin of replication. And this method involves taking advantage of the ability of dCas9. dCas9 is a catalytically inactive version of Cas9 that is able to recognize a double-stranded DNA sequence and bind to it, but it will not cleave double-stranded DNA. So you can add a fluorescent label to dCas9 and design a guide RNA to target whatever sequence you want in the genome, and you can get fluorescence. You can modify this idea by modifying a conventional guide RNA and adding another piece of RNA called MS2. And by introducing another protein into the system called MCP, MCP is a bacteriophage protein that is known to recognize MS2 RNA. If you add a fluorescent label to that, you can really enhance the fluorescence visualization of this dCas9 DNA interaction. What we hope to do is to use this system to visualize DNA replication, not at the origin of replication, but at a gene 0907, which is a non-essential gene, and it's not too far from the origin of replication. So this is something that I've been working on as an alternative. And because we have access to microscopy, because we have access to microscopes that are such high resolution, whether we look directly at the origin of replication or somewhere relatively close, it might not make a difference in this particular case. Another thing that I've been trying to work on, which is adapted from studies that have been done in E. coli, is to incorporate random fragments of M. cherry into DNA using transposon mutagenesis, putting that into the minimal cell, and screening for viable clones that are fluorescent, and then sequencing those clones to see where M. cherry might be functional. Next, I'm going to tell you a little bit about HUP-A, which is this bacterial histone-like protein that I talked about a little bit earlier. This histone-like protein, the expression of this histone-like protein has been significantly reduced in our minimal cell, and we think that is interesting for a lot of different reasons. So one way of looking at chromatin structure in bacteria and in other organisms is this method called 3C mapping, chromosome conformation capture. This is an experiment where you cross-link DNA with formaldehyde, and you can separate the cross-linked fragments from the non-cross-linked fragments with a restriction digest and some other methods. And then you can sequence those fragments. What you get is a two-dimensional plot with the diagonal line that represents the chromosome. And this fuzziness that you see around the line are representative of chromosome contacts within a bacterial genome. So this has been done in E. coli. E. coli, an actively replicating E. coli culture has about 20,000 Hub-A proteins in a cell. And what you can see here is that there's points of contacts all throughout the chromosome. When this experiment was done with SYN3A, the data suggests that there's no interactions along the chromosome. So E. coli has about 20,000 Hub-A proteins. JCVI SYN 1.0 has about 6,000, and that number has been significantly reduced to 28. When you look at the genome of SYN 1.0 in comparison to SYN3A, upstream of Hub-A in SYN 1 is this gene called GPSA. That gene was removed during the minimization process to generate SYN3A. What we want to do is add GPSA back into the minimal cell using CRISPR, transplant that into a capicolum cell, and then repeat this 3C mapping experiment to see if we can restore chromosome interactions after addition of GPSA. We also want to delete Hub-A from the minimal cell and see if that has any consequences on the ability of the cell to make these chromosome contacts. By significantly reducing Hub-A expression, we think that we have created a cell that resembles a primordial cell that emerged early in the evolution of life, specifically at a point where cells did not have sophisticated ways of organizing their chromatin. And by studying such a system, we think we can provide insight as to what those cells look like at that given time. Next, I'm going to talk about this transaldolase story. So our computational collaborators have developed a metabolic model for the minimal cell based on the genes that are present in the SYN3A genome and what we know about mycoplasma metabolism in general. And for the most part, we've been able to assign a gene to every single metabolic reaction across all major metabolic pathways. And this includes 155 genes. It's pretty much complete. And for the most part, it's consistent with the idea that the cell should double every 2 hours, with the exception of this enzyme called the transaldolase. So transaldolase is the enzyme that participates in the pentose phosphate pathway that produces nucleotide precursors. If there's any chemistry gurus in the chat, I have this reaction here for you. And the model predicts that the transaldolase is quasi-essential, meaning that the function of this gene can be disrupted, although at the expense of the growth rate of the bacteria. So it can be disrupted, but the cell will grow slower. To determine if the minimal cell makes a transaldolase, Andras Cook, who was a former research associate in the Glass Lab, what he did was take a well-characterized transaldolase from Bacillus subtilis, installed that into the landing pad of the minimal cell, and then subjected that cell to transposon bombardment. The idea is that if there is a transaldolase in the minimal cell that the model predicts is quasi-essential, adding a known transaldolase into the minimal cell would now make that quasi-essential gene non-essential. And after screening transposon mutants and doing some sequencing, he identified a potential candidate for transaldolase, although AlphaFold suggests that this looks nothing like a transaldolase. To determine if this is a transaldolase, what I'm currently working on is adding a histidine tag to this gene, purifying it from E. coli, and doing a quick biochemical experiment to determine whether this is a bona fide transaldolase. And if it's not a transaldolase, then we have to go back to the model and think about other ways in which the cell is able to carry out this certain type of metabolic reaction. And that's pretty much everything that I wanted to talk about today. I would like to thank everyone in the Glass Lab, Yo Suzuki's lab, who is also another lab at JCVI, and all of our collaborators. Thank you.
Ronald, you make me proud. Questions for Ronald? One thing I will mention, it is those, so with the labeled FTSZ, I was always concerned that while we can label it and cells that have the attached fluorescent protein are viable, there is still unlabeled FTSZ in the cells. And so it may be that the labeled FTSZ is not functioning to make a cell division ring. And so that's what you see sort of randomly distributed in the cells, and the unlabeled FTSZ still functions to make that. We probably ought to think about doing some experiments to find out if our labeled FTSZ can do what it's supposed to. We might have a conversation with Petrosvili about this.
Yeah, I think that's a good idea. Given that FTSZ is non-essential in a minimal cell, we can make a mutant of FTSZ. And if we can get some mutant phenotype in an FTSZ mutant, whatever that may be, it could be cell division, it could be cell shape, it could be anything compared to wild type, try to complement a mutant phenotype of FTSZ. And if we can complement a mutant phenotype with a fluorescently labeled FTSZ, I think that is enough evidence to say that a labeled FTSZ is functional in the minimal cell.
Possibly so, yeah. And then there are the Spilly lab in Munich has shown a great number of assays, in vitro assays, enzymatic assays, where you can look at FTSZ function. So we'll chat with her about that.